In this video, we'll be uncovering the differences between mitosis and meiosis. Mitosis describes the process by which the nucleus of a cell divides to create two new daughter cells, each containing an identical copy of DNA. Mitosis is important because it allows for asexual growth, maturation, and replacement of damaged cells. Meiosis is the process by which gametes are made in order for sexual reproduction to occur. It is the key component in sexual reproduction, and it adds to genetic diversity. Interphase is the period between cell divisions. During this time, chromosomes replicate and are loosely packed and not visible with a microscope. Two pairs of centrioles lie just outside the nucleus next to each other. A centriole is a cylindrical structure within the cell that plays a part in cell reproduction. Interphase is the same in mitosis and meiosis. Prophase starts at the beginning of the mitosis cycle, and prophase 1 of meiosis starts the first round of divisions in meiosis, where the goal is to separate homologous chromosomes. In prophase of mitosis, the chromosomes begin to condense. Each chromosome is actually two identical copies. Each copy is called a chromatid. Spindle fibers begin to form from the centrioles as they start to separate. Also, the nuclear envelope fragments and disperses. In prophase 1 of meiosis, the activities are the same as in mitosis, except a gene swapping process called crossing over occurs. The purpose of crossing over is to mix genetic material from the mother and the father. During prometaphase, the centrioles have moved to opposite ends of the cell, and the spindle fibers from both of the centrioles attach to each one of the chromosomes at the kinetochore. In prometaphase 1, the same thing happens as in mitosis, except the spindle fibers from each centriole attach to one chromosome of a matching chromosome pair. In metaphase, the chromosomes line up on the metaphase plate, and the spindle fibers start to pull each chromosome towards opposite ends of the cell. In metaphase 1, chromosome pairs called tetrads line up on the metaphase plate. In anaphase, the fibers pull the chromatids towards opposite ends of the cell. In anaphase 1, the tetrads separate. In telophase, the chromatids have now moved to opposite ends of the cell, and new nuclear membranes are formed. This completes the process of mitosis. In telophase 1, the chromosomes arrive at opposite ends of the cell. Though the cycle of mitosis is complete, the second round of divisions for meiosis is continuing. The purpose of the second round of divisions is to separate sister chromatids. In prophase 2, the same things happen as in prophase 1, except during the second round of prophase, crossing over does not occur. In prometaphase 2, the spindle fibers attach to the chromosomes at the kinetochore. In metaphase 2, the chromosomes align along the metaphase plate, and fibers from the centrioles begin to pull on each one of the chromosomes from both directions. During anaphase 2, the spindle fibers pull the chromatids apart and towards opposite ends of the cell. In telophase 2, the chromatids arrive at either end of each cell, and a new nuclear membrane forms. Telophase 2 completes the process of meiosis. Cytokinesis is the final cycle for both mitosis and meiosis. During cytokinesis, the cell cytoplasm is fully divided. This process is completed using a cleavage furrow in animal cells and a cell plate in plant cells. The resulting cells of mitosis are two diploid daughter cells that are identical to the parent cell. And the resulting cells of meiosis are four haploid daughter cells that are not genetically identical to the parent cell.